Welcome back. This is day 7 of genetics, and today we're going to be figuring out how can we analyze our DNA. But before we get to that, you guys have got 2 do not questions, so go ahead and pause this video, answer questions 1 and 2, and press play when you're ready to review them. So number 1, experiments revealed the following information about a certain molecule. We know that it can be broken down into amino acids, it can break down organic molecules into their subunits, and its function is severely reduced if it is heated or exposed to high or low pH conditions. So what is this molecule? Well, you should have gotten that this molecule is an enzyme. Now, I also asked you to define what exactly is an enzyme. So you probably had to dust off that Unit 2 vocabulary to write down that an enzyme is a biological catalyst that controls the rate of reactions. Specifically chemical reactions. All right. So let's get started. Now, I just told you that today we're going to be figuring out how can we analyze our DNA. But the first thing I want to ask you is, why is this important? So right now I want you to brainstorm, why do you think scientists might want to be able to compare the sequences of DNA between 2 people? So pause this video, answer that question, and press play when you're ready to review it. All right. Now, there are a few different options of answers that you could have set here. I'm going to focus on 3 that I think are the most important. So one reason we might want to compare sequences of DNA between 2 people is something like a paternity test, right? We can compare the DNA samples between a baby and possible fathers to figure out who the dad is, right? Same thing, we can look at family relationships. So if you have someone who was adopted and wants to figure out who their biological parents are, we can use DNA testing to figure that out as well. All right, number 2, you can also use DNA testing to solve crimes. If there was DNA found at a crime scene, we can use DNA testing to figure out or to test possible suspects and figure out who committed the crime. And now the third thing I'm going to mention, I know that this question was asking about the DNA samples between 2 people, but I'm going to apply that to just overall species as a whole, because we can also use DNA testing to look at evolutionary relationships between species. Right, so we can compare DNA of different species to figure out if they are closely related or not, and whether or not they share a common ancestor. So we'll be touching more on that in our next unit about evolution. All right, now if you said any other options that I maybe didn't write down, send me a message on Remind and we can figure out if that is a possible answer or not. It probably is. But now let's figure out how we do this. So we know it's important to be able to analyze DNA sequences, but how do we do that in a laboratory? Well, we have to use something called gel electrophoresis. And this is a procedure which separates DNA according to size using an electrically charged gel. So please write down this definition on your paper. So gel electrophoresis is a procedure which separates DNA according to size using an electrically charged gel. And this is a picture of what that actually looks like. So this blue section right here is that gel that we're talking about, okay? And it contains different wells where we're going to place the DNA samples. But the most important part about this is it has to be plugged up to a power supply. We have to have electricity in order to allow these DNA fragments to move down the gel, which I'll show you guys what that looks like in just a second. Right? So this is a picture of what is going to happen once we plug in our gel electrophoresis, okay? When you plug the gel into electricity, it's going to cause these fragments to move down the gel towards this end right here, towards the positive end. And those DNA fragments are going to separate according to size. So these bands that we see here at the top, closest to the wells that we place the DNA in, are considered longer bands. They don't move as much. Right? So they are longer or larger bands, but the shorter bands actually move quite a long distance. In fact, this right here would be our shortest band, right? Because it has moved all the way down the gel. So shorter bands actually move farther down the gel. We're going to look at a reason why in just a second. So the first thing I want to do with you guys is go through the different steps of gel electrophoresis. So each of these steps has to happen in the correct order in order for this process to work. And the first thing that we have to do is we actually have to cut up our DNA sample into fragments using an enzyme called a restriction enzyme. Now, we usually represent this enzyme as little molecular scissors because it's cutting up the DNA fragments. But this enzyme is not exactly like an Edward Scissorhands character running around, cutting everyone up. No, it is very technical in what it does. So it actually recognizes and cuts specific sequences of DNA. So it has like one job that it does. It knows, okay, I'm gonna cut just between the G and the C, or I'm gonna cut just between the G and the G. So it's very specific in what it does. But we have to use those restriction enzymes to cut up our DNA sample in order to allow it to move down the gel. All right, so then we're going to pipette those DNA fragments into this sponge-like gel. Again, these wells are where you're going to place the different DNA samples. So you're maybe going to put sample A right here, sample B here, C, and then D, so you can compare the different samples over time after they've moved. So these samples would be from different people. So maybe you would put the baby's sample right here, and then the father's samples here. Okay. The next thing that has to happen is you have to apply electricity to the gel to make one side of the gel negative and the other side positive. So this end is always going to be negative, where you first place the wells or the DNA samples, and this side is always going to be positive. Right? And this is what allows those DNA samples to move. But why do they move? Well, the reason they move is because DNA naturally has a negative charge, which means it's going to be naturally pulled towards, oh, what did I do? Hold on. Which means it's going to be naturally pulled towards the positive side because opposites attract. So because DNA has a negative charge, it's going to be pulled towards this positive end. Right, and this is where the size of the DNA fragments becomes important, because as those molecules move, the ones that travel the farthest are going to be the smallest fragments, right? And the smallest fragments actually move faster as well. Right? So smaller fragments are going to end up over here. While larger fragments stay over here. Right, and the larger fragments move slower as well. So as the DNA fragments get trapped, they form visible bands, and these bands are what actually allow us to compare the DNA sequences between different samples. So I'll show you guys what that looks like. Right here is a drawing or an animation of what this is going to look like over time. So again, those DNA samples move towards the positive end. Right now, I want you to tell me which one of these molecules is the smallest fragment. This right here is going to be our smallest fragment, right? It has traveled the farthest distance and it's also moved the fastest. But now, where are our larger fragments again? Well, these are going to be our larger fragments right here. They did not move quite as far. They move a little bit slower and they got stuck pretty early on. So everything in between is going to be pretty much just a medium-sized fragment, right? But if we were using this sample to compare maybe our baby right here to possible fathers, which one do you think is the dad? Well, that leads us straight to our fifth characteristic of gel electrophoresis, which is if DNA fragments from two samples settle in the same place, it indicates a relationship. So again, if this is our baby sample right here, and these are possible fathers, you're going to look for similarities between the two samples. So looking at our similarities, I actually see that these samples are pretty much identical to this baby's sample right here, right? We see that they have this fragment in common, etc., right? So those similarities indicate a relationship, which would mean that this guy is the father. All right. Now I've got some practice questions for you to go over. So now, I cannot speak today, guys. I don't know what's happening to me. So let's bring back our restriction enzyme. The first one we're going to be working with is something called HAE3. And HAE3 cuts the DNA specifically between the bases G and C. So on your paper, I've given you a DNA fragment or a DNA strand. I want you to figure out where would this enzyme cut the DNA sequence below. So if it's only going to cut between the bases G and C, where would it cut? And now, how many DNA fragments would be produced? Well, you should have gotten that enzyme is going to cut right here between G and C, and right here between G and C. Now imagine this strand is like a giant piece of paper. We've just cut it in half, or not in half, but we've cut it up. So how many different pieces or fragments are we going to have? Well, there's going to be one fragment right here, a second fragment right here, and a third fragment right here. So overall, this would result in 3 DNA fragments or 3 DNA pieces that have been cut up. Right. So now, once we've cut up that DNA, we're then going to place it in our gel. But if we were to place that DNA strand in our gel and turn on the electricity, how many different fragments would we see if we ran it through the gel? Again, we cut it right here and we cut it right here. So how many fragments would we see? Well, we're going to again have 3 DNA fragments in the gel, right? So this is a picture, not exactly the same picture as what we're dealing with, but if we were to place our sample in this well right here, over time, it's going to move toward the positive end, right, with the largest up here at the negative side and the smallest at the positive, which means that we would separate 3 of those DNA fragments. All right, one last question for you guys. So the diagram on the left illustrates a gel electrophoresis test comparing DNA fragments from 3 men and a baby. So we've got a paternity test here. Based on the results of the test, which man is the baby's father and how do you know? So right here, we've got the baby, and here we've got 3 possible fathers. So look at where those samples are similar and figure out who is the dad. Well, if you look at these fragments, you'll see that man 3 and the baby actually have identical banding patterns, right? So we know that man 3 is the father because they have identical DNA banding patterns. So I'm sorry to say, but man 3, you are the father. All right, guys, that's all I have for you today. So go ahead and complete your exit ticket, submit this on Google Classroom, and have a wonderful afternoon. I'll see you guys soon.